All right, hi everyone. This is going to be a quick review video for Exam 1 for Biological Psychology at Mississippi State. So I have the Canvas page pulled up here. This is for my Section 1, but it would look the same for my Section 2. So I just want to point out a couple of things here. One, the study guide that I'm going to pull up in a second is posted for you in Canvas. If you go to Modules, you see Exam 1 Study Guide, you can click on that. Also, there is an extra credit assignment for you guys as well. It is going to be closing right before the exam starts for your section. So for my section 1, it's going to be closing at 9:35 because that's when we're going to start taking exam 1. For my section 2, it would close at 11:10 when you guys would be starting to take exam 1. Okay, so with the extra credit assignment, there are 3 questions. You can answer all of them or just 1 or 2 of them and get some extra credit, as well as using this to help you study. Okay, so I've got the study guide pulled up here. The idea behind the study guide is that it helps direct your studying so you can see which topics you need to focus on. So when you see topics on the study guide, you want to go back to your notes to what we discussed in lecture. So exam 1 is going to cover everything that we've talked about thus far in the class, which would be chapters 1, 2, and 3. One thing that you'll see, and we'll get to this in a second, but I did highlight this section here on action potential. The reason that this is highlighted is because this is where the fill in the blank questions are going to come from. So the exam is going to have technically 51 questions because there's an extra credit question. So 50 required questions, and those will be the breakdown will be about 30 multiple choice questions, 10 true false, and 10 fill in the blanks. So the multiple choice and true false questions could come from any of these sections, but the fill in the blank is going to pretty much cover this process of action potential. Okay, so my goal here is to go through and just hit the highlights. You can also watch the lecture videos if you would like to. If there's anything that's confusing, you want to go back and listen to it again, or if you're missing any notes. If you go to the Canvas page for the class and click on the WebEx tab, then there would be a tab here that says recordings, and there you should be able to find the lectures that we've done so far. All right, so if we hit the highlights before exam 1, chapter 1 had 2 major topics. Primarily, we focused on research methods, and there was a little bit about history as well. So when we talk about the 5 research perspectives, different ways of doing research in biological psychology, we talked about describing behavior. We talked about comparing behaviors in different species. So that was the sort of the evolutionary view there about if species are similar. Is that because they have a similar ancestor, or maybe because they developed in a similar environment, that sort of thing. We could also study development, so we could look at one individual throughout the lifespan. We talked about studying mechanisms, so trying to figure out exactly which part of the brain is involved in speech, for example. And then applications. Can we find ways to use what we've learned to help someone who has some sort of disorder or disability, etc. All right. With research methods, we compared and contrasted correlational and experimental research. Both of these are valid types of research, but the question is, what is the purpose? So the purpose of correlational research is for prediction, and the purpose of experimental research is for determining causation, so understanding why something happened. So with correlational research, you take 2 naturally occurring variables, measure them, and look for a relationship between them. You don't actually manipulate anything, like measuring the amount of sleep someone has gotten at night and their mood the next day. I didn't manipulate your sleep or manipulate your mood. I just measured them and looked for a relationship between them. Now, with a correlation, if you know the relationship between 2 variables, you can use one to predict the other. So if I know the relationship between sleep and mood, I can use your sleep to predict your mood, or your mood to predict your sleep. What I can't do is determine causation here. So in order to determine causation to be able to answer the why question, and by the way, I'm at home recording. My husband's in another room with my kids, but like real life is happening right now. So just be advised, there's no telling what you might hear in the background. With experimental research, you're actually going to be manipulating something. So, like, for example, I could manipulate the amount of sleep some people get, which just seems cruel, but something my toddler's been doing for a while now, where you intentionally keep a person awake and then you look to see how that impacts their mood. So here you're actually manipulating something. So one other thing to say about correlational research is that we have direction and strength. So with a positive correlation, the two variables are moving in the same direction. Sleep goes up, mood goes up, sleep goes down, mood goes down. That's a positive correlation. With a negative correlation, you have variables that are moving in opposite directions. So as one variable goes up, the other variable goes down. As one variable goes down, the other variable goes up. So they're moving in opposite directions, right? Absences goes up, grades go down. Absences goes down, grades go up, that kind of thing. And then also the strength. When you're determining the strength of a correlation, you just look at the number. The number would be anywhere between 0 and 1, with 1 being a perfect correlation and 0 being no correlation at all. The bigger the number, the closer it is to 1, the stronger the correlation, the better your prediction. So a 0.6 correlation is stronger than a 0.4 correlation, for example. But there you would not look at the sign. So a negative 0.5 and a positive 0.5 correlation would have the same strength because they have the same number. Okay. With an experiment, we are actually going to be manipulating something. So we talked about two different types of interventions. With an intervention, we're intervening. This is an experiment. We're manipulating something. With a somatic intervention, you're manipulating something biological, and you look to see if there's a change in behavior. So here the independent variable would be something biological, and the dependent variable would be something behavioral. With a behavioral intervention, we're changing something in the environment or something related to behavior to see if there's a biological outcome, a biological change. So here the independent variable would be something behavioral or environmental. The dependent variable would be something biological. Remember that the independent variable is the variable that the researcher is manipulating. The dependent variable is the variable that the researcher is measuring. So for a somatic intervention, I could give a group of individuals a medication, and I could look to see if it changed their social behaviors or their sleeping behaviors or whatever the case might be. With behavioral interventions, maybe I could have a group of individuals go to therapy, and then I could measure their serotonin levels afterwards. Okay. Let me know if you have questions also as you're studying. You can ask me in class on Tuesday or email me as well. Okay, as far as the prevalence of neurological and psychological disorders go, I'm not going to expect you to know any numbers. Remember, we had these graphs in the slides. Know a little bit about which ones are most common. So, for example, with psychological disorders, the most common ones would be anxiety disorders. And we talked about how that might be because there are several different disorders that fall in the anxiety disorder category, or maybe because there's less of a stigma related to getting help for an anxiety disorder versus other types of disorders. And then briefly, we talked about the history of biological psychology, how early cultures thought that the heart was most important for cognition, for emotion, etc. And you can see that in the way, like, for example, the way Egyptians made mummies, the way they treated the heart versus the way they treated the brain. Later cultures, we started to realize the importance of the brain, but there was still a long way to go as far as understanding the way that the brain worked. Phrenology was the idea that you could feel a person's skull, like literally examine their skull, and that the shape of the skull would tell you something about their abilities or their personalities. Like, hey, this person has a bump right here. This must be a creative person because that's a creative part of the brain. There is some truth here in that localization of function is true. There are certain parts of the brain excuse me, that handle certain functions, but you can't tell that by feeling someone's skull. It's not as if that part of the brain is so much more developed that it causes a literal bump in the skull. So there was some truth, but a lot of misinformation in phrenology. Okay, chapter 2, we looked at the anatomy of the nervous system. We started off looking at the cellular level, and then we backed up and looked at gross neuroanatomy, so the parts of the nervous system that you could see without a microscope. So When it talks here about neurons, some of this information is consistent across types of neurons. Like, for example, the fact that you have a message reaching the axon terminals and causing a neurotransmitter to be released and crossing that synapse and then binding to the dendrites on the next neuron, like that's consistent across neurons. There are some things that are different from neuron to neuron. So one thing we said was that the most common type of neuron by shape is multipolar. The most common type of neuron by function is an interneuron. So that's one thing here. I have this picture pulled up here from the slides. This is in chapter 2. So we talked about 3 different shapes of neurons, and multipolar neurons, as I said, are the most common by shape and typically what we're thinking about when we're thinking about a neuron. One thing to note is that a unipolar neuron is a little bit different. The cell body is a little bit lower down, so it's not part of the input zone. So the general idea here is that messages come in the input zone, which would always include the dendrites and sometimes the cell body. You see for multipolar and for bipolar, the cell body is in the input zone. So we always had messages coming in. The dendrites could also come in through the cell body as well. All of those messages are integrated in the integration zone, which is typically the axon hillock. This is where the cell body is tapering into the axon. You can see that here. I say typically because, once again, the unipolar is a little bit different. Here we have our integration zone here, and we have our axon hillock down lower. So not always, but typically the axon hillock is the integration zone. If the message is passed, if the neuron fires, it will start at the axon hillock and go down the conduction zone, which is the axon. You see that the axon is coated in a myelin sheath. That increases the speed of transmission. So the more myelination you have there, the faster the message is conducted. Then you reach the output zone. So this would be where the axon branches out into axon terminals. When the message reaches the axon terminals, that causes neurotransmitters to be released. So we have neurotransmitters that are stored in synaptic vesicles inside the axon terminals. And when the message reaches the axon terminals, that causes neurotransmitters to be released into the synapse. They diffuse across and bind to receptors on the next neuron in the next neuron's input zone, which could be dendrites, or for some neurons could also include the cell body as well. Okay, so let's see. So the presynaptic membrane is going to be the axon terminals. And where's my picture? Here's my picture. This is the presynaptic membrane because this is where the message is going to be released into the synapse. Then you have a synapse, a small gap in between, and then you have the postsynaptic membrane, which would be the input zone of the next neuron, which would always include the dendrites and sometimes the cell bodies. I have a picture here. So here we have the presynaptic membrane. This is an axon terminal. Here we have the synapse, the gap in between, and then you have the postsynaptic membrane. So on the other side of the synapse, you have what could be a dendrite. This is a dendritic spine in this picture, or it could possibly be a receptor on a cell body as well. Okay, yes, good. The thing about axonal transport is that the neurotransmitters and synaptic vesicles and things that are needed in the axon terminals are actually put together in the cell body. And then they're transported from the cell body down to the axon terminals. And they do that by going down the middle of the axon. So the axon is sort of like a hollow tube. The electrical message that's being passed, that's happening on the outside of the axon. And with axonal transport on the inside of the axon, you have finished materials headed from the cell body down to the axon terminals, but then you also have recycled materials going from the axon terminals back towards the cell body so they can be recycled. Okay, we talked about 4 types of glial cells. We said that astrocytes help control the flow of resources, so giving extra oxygen to the neurons that are most active, for example. Of course, you have to be careful there because anything that's impacting the activity of neurons, if we have too much activity, that can trigger a seizure. So astrocytes could be connected with epilepsy in some situations. We talked about microglial cells, small glial cells that clean up debris and how they could be connected to something like Alzheimer's disease, for example. We have these plaques that have built up, and either there's so many plaques that the microglial cells can't handle it, or the microglial cells are just not doing their job well. And then we talked about oligodendrocytes and Schwann cells. Both of these are involved in myelination of axons, so wrapping axons in that fatty tissue, that myelin sheath. Remember that the oligodendrocytes get to go into the brain, so they're going to handle that myelination for the central nervous system, and Schwann cells handle that myelination for the peripheral nervous system. Okay, so when we break down the nervous system, we have these different divisions here, and there is a graph in your slides. I don't have chapter 3 pulled up here. Is it in chapter 3? No, it's not. It's in chapter 2, so I do have it. Hang on. Hang on. Here we go. This is in your slides. This is part of the chapter 2 slides. This can be helpful as you're looking at the subdivisions here as well. So you might refer back to this graph as you're studying. So the central nervous system breaks down into, or I should say the nervous system, there we go, breaks down into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord, and the peripheral nervous system consists of those nerves that are connecting the brain and spinal cord to the rest of the body. So the peripheral nervous system breaks down into the somatic and autonomic nervous systems. Somatic nervous system would handle motor and sensory functions. So this would be, your brain is sending out messages to your muscles to move them, or things in the environment are sending in messages into your brain. So the cranial nerves handle the motor and sensory messages for the head and neck area, and the spinal nerves carry the motor and sensory messages for the rest of the body. The autonomic nervous system handles more automatic sorts of things. This is something like increasing or decreasing your heart rate, increasing or decreasing digestion, dilating or constricting your pupils, that sort of thing. Now, even I left out enteric here on the study guide. The autonomic nervous system has 3 components: sympathetic, parasympathetic, and enteric, but we just really didn't talk about that much. But the enteric nervous system would also go under autonomic, and that's only focused on specific like gut functions. But the sympathetic nervous system is your fight or flight system, which would like increase heart rate, increase respiration rate. It stops digestion so that your resources can go towards life-saving functions like escaping from danger. Versus the parasympathetic, this is your resting and digesting, so heart rate decreases, breathing slows down, digestion increases, that kind of thing. We do need a balance between the sympathetic and the parasympathetic. Okay, the central nervous system consists of the brain and spinal cord. When we're breaking down the brain, this is where I really feel like this figure in your slides can be really helpful for you here. We talked about how in the beginning we have a neural tube and how it's important for a pregnant individual to get enough folic acid. That's a B vitamin that can help prevent some neural tube defects. We can't prevent all of them, but that would be helpful. How the neural tube has really 3 sections: the forebrain, the midbrain, and the hindbrain. With the most accurate way of thinking about this, that the forebrain is towards the top, and then the midbrain in the middle, and the hindbrain at the bottom, as opposed to thinking about the forebrain being in the front and the hindbrain being in the back. That's not really the best way to think about it. So you have this in your notes, but the forebrain breaks down into the telencephalon and the diencephalon. And then we have the midbrain, and then the hindbrain breaks down into the metencephalon and the myelencephalon. So in the forebrain, the telencephalon consists of the cerebral cortex, the basal ganglia, and the limbic system. We said that the cerebral cortex has 4 lobes. You have 1 lobe on each side. So you have your left temporal lobe, right temporal lobe, for example. You know, the frontal lobes in the front, which handle things like personality. A large portion of attention happens in the frontal lobe, thought, decision-making, that kind of thing. The occipital lobe's in the back, and that is pretty specialized for vision. The temporal lobes are on the side towards the bottom, sort of around closer to your ears, and they're going to handle auditory information, but then also be involved in memory, emotions. And then the parietal lobe would be sort of up above the temporal lobe, and the parietal lobe, while it processes a lot of things, we primarily think about touch happening there. When it talks about the boundaries between them, we talked about the terms like gyrus and sulcus, that a gyrus is a raised part of a fold on the cerebral cortex, and a sulcus is a lower part, and how the brain is actually fairly standardized, so we can use those different gyri or sulci, I'm going to use a Latin pronunciation there for that, to see where we are in the brain. That's the cerebral cortex that's in the telencephalon. Also in the telencephalon, we have the basal ganglia. Basal ganglia involve structures that are important for movement. We said that this is where the terminology is not completely accurate here because we are in the central nervous system. We shouldn't be using the term ganglia. That's a peripheral nervous system term, but this is what they're called, so we're going to stick with that. The basal ganglia are most connected with movement, and we talked about how most of these structures are in the forebrain, right? So the caudate nucleus, the putamen, the globus pallidus, they're in the forebrain, but how the substantia nigra is actually technically located in the midbrain, but because it's connected with those forebrain structures, it's considered part of the basal ganglia. The limbic system, still in the telencephalon here. The limbic system is involved with emotions and with memory. So we talked about like the amygdala, for example, and how important that is for processing, especially negative emotions like fear. And we talked about the hippocampus, mammillary bodies there that are part of the memory-related functions in the limbic system. Okay, the diencephalon, we're still in the forebrain, but if we move away from the telencephalon to the diencephalon, here we have the thalamus and the hypothalamus. The thalamus is sort of a relay station for sensory information. Almost all sensory information goes through the thalamus, with smell being the only exception, so that information doesn't go through, but all the other senses, that information goes through the thalamus, and then it's routed to the part of the cortex that's going to process that information. So if it's visual, it's sent to the occipital lobe. If it's auditory, it's sent to the temporal lobe, that kind of thing. Okay, and then we have the midbrain. I don't see anything on here about the midbrain, but there are midbrain structures that are important, like the substantia nigra, for example, for movement. We talked about the superior and inferior colliculi that are important for sensory functions. But then if we move into the hindbrain, we've got the cerebellum and the pons and the medulla. The cerebellum is called the little brain. It sort of looks like a smaller version of the brain. It's got the convolutions, the folds. The cerebellum is important for coordinated movement as well as for learning. So that's kind of newer information, but we know that like classical conditioning requires the cerebellum. And then the pons and the medulla. Here we're getting to a point where we're sort of starting to taper off into the spinal cord. We're at the very bottom of the brain. So these are important for some very basic functions. The pons is involved in movement. It's involved in sleep as well. We'll talk later in the class about how the pons is related to REM sleep. The medulla is important for like regulating heart rate and respiration rate, so really important there. We have layers of protection in the brain. We call the meninges is the term that we would use here, and we have 3 layers. You have the, remember we're going to go with the Latin here, so the pia mater, the tender mother. So there's that layer that's right on top of the brain that's very thin. And then the outer layer is the dura mater, it's a tough mother, right? In between we have the arachnoid, which is sort of an open space where you have cerebrospinal fluid, but there are some almost spider web-like projections in there, and so that's why it's called the arachnoid. So we have the cerebrospinal fluid there that is protecting the brain, acting as a shock absorber and also as an exchange medium so that things that we need from the blood, like oxygen and nutrients, can get into the brain, and then things that we need to get rid of, debris, etc., can leave the brain through the bloodstream. Okay, chapter 3. This is where the action potential process is discussed, and this is where the fill-in-the-blank questions are going to come from. So when we talked about the action potential process, we started off talking about what a neuron is like at rest. So with our neuron at rest, it has a negative charge. If you measure the fluid inside the cell and compare that with the fluid outside of the cell, at rest, the intracellular fluid has about a negative 65 millivolt charge. So it's about 65 millivolts more negative than the surrounding fluid outside the cell. Also, at rest, the sodium channels are closed and the potassium channels are open. So we have these ion channels that are embedded in the cell wall that can determine what comes and goes. They're selectively permeable, so if it's a potassium channel, it's only going to let potassium pass, for example. So at rest, we have negative 65 millivolt charge. Our sodium channels are closed, our potassium channels are open, and the sodium potassium pump is working. It is a pump, so it does require energy. It's going to be pumping out 3 sodiums for every 2 potassiums that are pumped in. Remember that the abbreviation for sodium is Na, and for potassium it's K, and that these have a plus sign there because they're cations. They're positively charged. You can remember that a cation is positively charged because it has a T that looks like a plus, versus an anion, which is like a negative ion. It's a negatively charged ion. Okay, now we talked about how there are different forces that are impacting the flow of ions. We have diffusion and electrostatic pressure. Diffusion is just the idea that the molecules will want to spread out. They'll want to move from an area of high concentration to an area of lower concentration until they're equally spread out throughout the available space. Electrostatic pressure is the idea that like charges repel and opposite charges attract. Talked about the sodium potassium pump. Okay, so with action potentials, here's what's happening. You have messages that are coming in through the dendrites. Let me pull up this picture again. That's not what I'm looking for. Here we go. You have messages that are coming in always through the dendrites and possibly through the cell body, depending on which type of neuron we're talking about. So you have messages coming in the input zone. Some of these messages will be excitatory and some of these messages will be inhibitory. The excitatory messages will push the neuron closer to threshold, closer to firing. The inhibitory messages will calm down the neuron. They'll make it less likely to fire. They'll push it further away from our threshold. So at rest, we're at negative 65. You have messages that are coming in. If it's a hyperpolarizing message, it's an inhibitory postsynaptic potential, then it causes our neuron to go from negative 65 to negative 70 to negative 75. We're getting further away from threshold, less likely to fire. If we have excitatory messages coming in, then that's going to depolarize our neuron. We're going to go from negative 65 to negative 60 to negative 55. We're getting closer and closer to threshold. If we reach threshold at the axon hillock, that causes the action potential to start. So the average resting membrane potential is about negative 65. That's where we are at rest. The average threshold for an action potential is about negative 40. So we have to depolarize from negative 65 to negative 40. If we do, that causes an action potential to occur. The action potential will start at the axon hillock, and then it will be propagated down the axon. So we have one section has an action potential, and the next section has an action potential, and so on and so forth. And it's going to be flowing from the axon hillock towards the axon terminals and not going back the other way because of the refractory period of the previous section. If this section has just had its action potential, now it's in a refractory period. It's not firing again right now, which keeps the message going in the appropriate direction. Okay. So what actually happens when action potential occurs? So we depolarize from about negative 65 at rest to about negative 40 as our threshold. When that happens, the voltage-gated sodium channels open. Up until this point, the sodium channels have been closed, so sodium has not been able to come and go freely. And sodium has been pumped out of the cell by the sodium-potassium pump. So we have sodium building up outside of the cell, and it can't get back in until we hit threshold, about negative 40. The voltage-gated sodium channels, those are sodium channels that only open when we have a particular change in charge. That's the voltage-gated bit. So when the charge changes to -40, that opens the sodium channels. Sodium is going to come rushing in because A, diffusion, it's been building up outside of the cell, and it doesn't want to be all clumped up on top of itself. It would rather spread out. So part of that is diffusion. It wants to spread out. It spreads out into the cell now that it can, now that the sodium channels are open. Also, part of this is electrostatic pressure. Sodium is a cation. It's positively charged. The interior of the cell is negatively charged. At this point, we have hit -40. So that's still negatively charged. Not as negatively charged as it was at rest, but still negatively charged. And that will draw in the sodium. So the sodium channels open at -40. Sodium rushes in and the sodium channels close. Now the inside of our cell at the height of an action potential is actually positively charged up to about plus 40 millivolts. At that point, the cell is not going to fire again. We're at plus 40. We are so far away from -40, it's not happening right now. So that's the absolute refractory period where we cannot have another action potential at that time. Then the cell starts working towards getting back to its resting state. And while it's working on getting back to its resting state, we're in a relative refractory period, which means that it could fire again, but it would be very difficult to get it to fire again. Once it gets back to its resting state, though, then we'll be back to being ready to fire again. How does it get back to its resting state? Well, at the height of action potential, sodium has rushed in, and now the charge inside of our neuron is positive 40. Well, the sodium's trapped because those sodium channels closed, but potassium is free to leave. The potassium channels are open pretty much all the time. And now potassium wants to leave because potassium is positively charged. At the moment, the interior of the neuron is also positively charged, so that will repel the potassium and the potassium rushes out. When the potassium rushes out, our cell goes back to being negatively charged. The only problem is we've got all of our sodium inside the cell and all of our potassium outside of the cell, and we'd like the opposite. And that's where the sodium-potassium pump starts working. It starts sending out 3 sodiums for every 2 potassiums that come in, and that helps the cell get back to its resting state. All right, that's a quick overview. If you have questions, let me know. Also, you can always go back and rewatch the lecture videos as well. Now, this last bit here we actually haven't done in class yet because this is what we're doing in class on Tuesday, but I wanted to go ahead and do this video and get it up for you. So we will talk about this and a little bit of other topics as well in class on Tuesday. So you'll see when we talk about the term ligand, we're talking about a substance that is going to bind to a receptor. And we're going to talk about the fact that there are 2 types. You have endogenous ligands and exogenous ligands. An endogenous ligand is something that your body is naturally producing. It's a ligand, so it's going to bind to a receptor, but it's an endogenous ligand. So this would be something like a neurotransmitter, for example. Your body's naturally producing something that's binding to a receptor. Versus exogenous, it's still a ligand, so it's still binding to a receptor, but the difference here is that this is something that's coming from the environment, so a drug, for example. You'll see also that when you have a ligand bind to a receptor, it can act as an antagonist or an agonist. We're going to go into a lot more detail on this in Chapter 4, but in Chapter 3 we're just going to lay the groundwork here. Then an antagonist would be a ligand that would bind to a receptor and block it. So if you have a GABA antagonist, for example, GABA is a neurotransmitter. If you have a substance that binds to a GABA receptor and acts as an antagonist, then A, it doesn't activate the receptor, so it doesn't do anything. But then B, it blocks the receptor so that GABA can't come and bind. So it's blocking the effects. An agonist is something that binds to a receptor and activates it. So GABA binds to GABA receptors and activates them. But there are also exogenous substances, drugs, that can also bind to a GABA receptor and activate them. So if it's activating the receptor, then it's an agonist. If it's just parking there and blocking it, that's an antagonist. And then when we talk about types of receptors, we'll discuss this in class on Tuesday. An ionotropic receptor is a fast receptor. So here you have a neurotransmitter or any kind of ligand binding to a receptor. If it's ionotropic, it immediately opens an ion channel that is right there in the receptor, which means that you can have cations or anions coming in immediately. So then, if we're talking about an ionotropic receptor, this is a quick effect. Immediately, we're opening the ion channels and the substances is coming in. With a metabotropic receptor, you have your ligand bind to that receptor, and then it causes a chain reaction that will open an ion channel. But the ion channel is not part of the receptor. It doesn't happen right away. So there are some substances that take a little bit longer to have their effect because they're activating metabotropic receptors. We have our ligand bind to a metabotropic receptor, and then it sends out a messenger G protein to open an ion channel so that eventually it will open and you will have cations or anions rush in, but it takes a little bit longer, so it's a little bit slower. And then also what happens when our neurotransmitters are done. Excuse me. All right, what happens when our neurotransmitter has been released from the presynaptic membrane, diffused across, bound to the postsynaptic membrane, had its effect, and now it's done? What do we do with the neurotransmitter? Well, our body wants to be efficient and wants to recycle and wants to reuse. So there are a couple of things that can happen here, and we'll go into more detail on Tuesday, but the general idea is that it can be taken back up whole. We can just take it back up, or we can break it down into smaller parts and then take it back up. Either way, we're going to be reusing our neurotransmitters here. And actually, there are a lot of drugs that interfere with this particular bit. So if you think about an SSRI, a selective serotonin reuptake inhibitor, it's blocking that reuptake of serotonin so that it stays in the synapse longer, so you can have more of an effect from it. So we'll talk about this in class on Tuesday, but a lot of the effects we have from substances are happening because they're impacting what happens to neurotransmitters. Okay, and then the last topic that's on the study guide here would be seizures. We will mention this in class on Tuesday. We're not going to discuss this a whole lot. We'll talk about how an EEG can be used to measure the electrical activity in the brain, and that with a seizure, we have some sort of synchronization of electrical activity that's not supposed to happen. It's happening all at one time. Now, there are many different types of seizures and lots of different causes for them. We will talk about kind of three major categories of seizures. So we'll talk about tonic-clonic, simple partial, and complex partial. The difference between tonic-clonic and the partial seizures would be how much of the brain is involved. So tonic-clonic is another term for a grand mal seizure or a generalized seizure. So that involves pretty much the entire brain, versus a partial seizure that only impacts certain parts of the brain. And then you'll see simple versus complex seizures. The simple versus complex seizures, we're talking about partial, so it's not involving the entire brain. The simple part would say that you don't lose consciousness. The complex part would have more of a loss of consciousness. So how aware is the individual during the seizure? That's part of what you can use to differentiate between simple and complex. But of course, it's very difficult to diagnose a type of seizure unless the individual is hooked up to an EEG while the seizure is happening. But that's just a little bit about what's happening. So with tonic-clonic, you have those periods of rigidity, and then you have those periods of movement. We'll talk about that in class, but that's previously been called a grand mal seizure. That would be generalized. It would involve the entire brain, versus partial seizures that only involve certain portions of the brain. The specific symptoms that a person has with a partial seizure will be different depending on the location of the seizure. Since it's partial, it's not involving the entire brain. But with a simple partial seizure, the person will retain consciousness during the seizure, versus with a complex seizure, the person might lose consciousness or have kind of an altered state of consciousness. So very confused, acting very strangely, that kind of thing. Okay. So we'll talk a little bit more about that in class on Tuesday. I hope that this review video is helpful for you. If you have questions as you're studying, please feel free to email me or ask me in class on Tuesday. Just let me know if you need anything. You guys take care.